In this video, we're going to cover the basics of DNA replication. Now, DNA replication is carried out by the enzyme DNA polymerase. And replication works by using a single-stranded DNA as a template. And from these videos, anytime you see ssDNA, that means single-stranded, dsDNA means double-stranded. And what happens is that you have a nucleophilic attack on the DNA chain at the 3' hydroxyl, so that's why it's always adding 5' to 3'. We need the 3' hydroxyl. And it's going to attack the alpha phosphate of a nucleoside triphosphate. The inorganic phosphate, or pyrophosphate rather, PPI, will be removed. And the removal of these phosphates is the energy that is needed to make this new covalent bond and to add this new base. Now, just about every single polymerase that we know of can only add to the 3' hydroxyl, so we're always adding to the 5', the 3' direction. Now we're going to start with E. coli replication because it's a lot easier to think about than eukaryotic replication. And during synthesis, you have eyes or bubbles being formed, and these are called theta structures, theta replication, and they kind of look like a theta. And at each branch point, you have two of these here, is a replication fork. And this is where DNA synthesis is occurring. And they have, or rather E. coli will replicate in both directions from the start of replication. So the replication forks will go in opposite directions of each other and will move apart while they are making a copy of the DNA. Now, since DNA can only be made in one direction, 5' to 3', it is semi-discontinuously synthesized. What that means is that the leading strand always has a 3' hydroxyl, so that just continues on and on and on. However, the other strand, the lagging strand, does not. So we need to make the lagging strand in fragments because I can only add to the 3'. So every time I unzip a little more, I have to make a new fragment. These fragments are called Okazaki fragments. Eukaryotes, the fragments are only 100 to 200 nucleotides, while prokaryote can have Okazaki fragments that are 1,000 to 2,000 nucleotides. And the polymerase itself can't join these Okazaki fragments. You need a ligase, a DNA ligase, that will connect these fragments together so you're going to have complete replication. Now, the 5' ends of all Okazaki fragments are RNA because a polymerase needs a primer, an RNA primer to start with, and we keep making new and new fragments. And so every time we want to synthesize a new Okazaki fragment, we need a primer there. Depending on the species, these primers can range from anywhere from 1 to 60 nucleotides. The enzyme that takes care of making these RNA primers in E. coli are called Primates, which is a nicely named enzyme, tells you exactly what it does. And the way this works for Primates is that RNA synthesis is initiated by linking two ribonucleotides together. And like I said, every time you make a new fragment, you need a new primer. However, for the leading strand, you only need the primer once because it's continuous. Once you start it, you don't need to stop it. But eventually, we don't want RNA in our DNA sequence, so these primers are removed before replication is complete. And that's it for this basic overview of replication. And in the next series of videos, we will dive into this topic more. Until then, see you next video.